Welcome to chapter 4.1. So we are starting unit 2 now. Unit 2 is just a 2 chapter unit, so it's going to cover chapters 4 and 5. So we're going to dive into chapter 4. Now, a note on chapter 4. So chapter 4 is kind of a review of biology, especially biology 201. So all of chapter 4 pretty much reviews the basics that we need for micro from chapter 2 of biology 201 to about chapter 5 of biology 201. So we're going to talk about the chemistry of life. We're going to talk about macromolecules, uh, transport, so endocytosis, exocytosis, and all that. So this should be, for the most part, a review, so you've seen it before, but we need to pay attention because we're going to tailor our knowledge from biology and we're going to look specifically at microbes here in microbiology and look at how these affect bacteria and small microbes like that. Okay, so chapter 4 objectives. Chapter 4 is called Living Chemistry from Atoms to Cells. The things that we will be able to do when we're done with chapter 4 are we'll be able to explain that cells consist of molecules, and we'll be able to describe the properties of those molecules. You'll be able to explain how a pathogen can alter a host cell's biochemistry. You'll be able to describe how membranes mediate or allow the transport of molecules and particles across it. And you need to be able to explain how pathogens can take advantage of transport processes. Okay, so let's talk about this chapter's case study. So our case study is called Lethal Chemistry from a Waterborne Pathogen. So when we talk about water, water is essential for all life on Earth. It's the most abundant molecule on Earth, but there's some downsides to water. Too much water can be deadly in a whole host of different ways. So what we need to do for this case study is we need to set the scene. Okay, so we can understand what's going on in this case study. So our case study takes place in Pakistan in the late summer of 2010. So Pakistan is experiencing very heavy rainfall during the summer. That's very unusual. Okay, in fact, there's so much rain falling, Pakistan is starting to flood. In fact, 1/5 of the whole entire country is under several feet of water. Okay, because of this flooding, millions of people have been displaced from their homes, but the water is also running in places that it normally wouldn't. So we have it running from the countryside into the cities. We have it going into the sewer systems and backing them up and contaminating the water with sewage. So this water becomes quickly contaminated with a whole host of different pathogens. And a lot of these pathogens, one that we're going to talk about specifically, are going to cause diarrheal disease. So these people that come down with this disease are going to have extreme diarrhea that's going to cause extreme dehydration. Okay, although a lot of the population has become sick, we're going to talk mainly about the child population of this country because they're going to be the ones that are most affected. Okay, we're going to talk about this in kind of a mechanistic way. So there are different mechanisms by which pathogens can cause disease, and we're going to talk about one of them. We don't talk a lot about the different mechanisms in this class, but we do we do talk about one or two of them, and one of the most important ones we're going to talk about today. Now, diarrheal disease will cause extreme dehydration, and it's actually very deadly. Diarrheal disease is going to cause watery stool, so you're going to have so much water exiting your system that you're going to become dehydrated very quickly. In fact, you are going to keep becoming more and more dehydrated, and you're going to reach a point where there's not enough water in your body to support your body's chemistry and to keep things running, so you actually die. Diarrheal disease is actually really hard to treat in scenarios like this case study because there's just no clean water around. Most of the water is contaminated, and getting clean water is a very difficult thing in these scenarios. And children are most vulnerable because they're really small. So teens and adults are considerably larger than children, which means they have a lot more water. So it takes a longer time for them to become dehydrated. Children, because they're small, they don't have a lot of water, so they get dehydrated really easy. So when we have cases of this diarrheal disease going through a community because of contaminated water, the children are going to be the most heavily affected because of their size. Okay, so let's talk about what's happening here. So we're going to follow a boy named Akal. Okay, so Akal has drank the water that's contaminated. So he is starting to have that diarrheal disease. He has profuse diarrhea. And because of that, he's starting to become extremely dehydrated. But there's just not any clean drinking water available, so he keeps drinking the contaminated water. So because of this, his diarrhea is extremely watery. Okay, and it's actually going to be filled with these white flecks of mucus. They're from the intestine because the water's going through his system so fast. It's actually starting to flood some of the intestinal mucus out of the intestinal tract and into his stool. So when we ever see cloudy diarrhea with white flecks, this is what we call rice water stool because it looks like little grains of rice in the water. Okay, so this is what kind of diarrhea Akal has. Because he has this extreme diarrhea, he's going into extreme dehydration, so he becomes listless, he doesn't have a lot of energy, his eyes are going to start to shrink and become sunken, okay, and his skin's going to start to wrinkle because all the water that keeps it nice and hot and filled is just not there. Okay, so because of this, Akal is taken to the nearest medical center, and this is a natural disaster. There's a lot of people at the medical center. They have all hands on deck. So Akal is seen by a nurse, and the nurse takes a sample of Akal's stool, and then he takes that sample and does the clinical microbiology on it. So he goes and looks at Akal's sample underneath the microscope. So one of the things he did is he used dark field microscopy. So this is like bright field, except it has a dark background. Okay, when he looks at Akal's stool, he observes that there's quite a few, aka a lot, of bacteria swimming around rapidly. In fact, he noted that the bacteria, because of the masses and how much there was, were like twinkling stars. Okay, so the bacteria that this nurse is seeing in Akal's sample is very distinctly shaped. Okay, it's curved, comma-shaped bacilli. So they're comma-shaped rods. Okay, when they did the gram stain on the sample, the bacteria gram stained negative, meaning they're going to appear pink. Now, if you have a background in medical microbiology, these are pretty well-known bacteria, so you'll be able to diagnose this really quickly. So with what he saw underneath the microscope and with later testing, Akal was confirmed to have a Vibrio cholerae infection. So Vibrio cholerae is the bacteria that causes cholera, which is a type of diarrheal disease. So Vibrio cholerae bacteria produce the cholera toxin. Okay, so it's kind of like a little protein that is going to be absorbed by your intestinal epithelial cells, so the lining of your intestines, and it's going to cause your intestines to release a lot of the chlorine 1- anion. Okay, so you're going to release a lot of chlorine ions from your cells, which are then going to go into the intestinal tract, and it's going to combine with the cations that are found in those, like sodium 1+, found in the intestinal tract. Well, we all know that positive and negatives are going to be attracted to each other. It's going to make a compound. It's going to make salt. Okay, so we are going to start to change the concentration gradient between the outside of the cell and the inside of the cell. If you remember back into biology, this is how we moved things across the membrane. Okay, so because we are making this salt in the intestinal tract outside of the cell, the concentration of salt is going to increase to a point where all the water from in the cell is actually going to rush out, trying to equalize this concentration gradient. Okay, but because the toxin is causing the cells to continuously leak chlorine, more and more salt is being made, more and more water is being lost from the cell, which is going to cause your watery diarrhea, and you just are going to start to be dehydrated because of it. So at this point, when a call is getting seen, he is in pretty dire straits. Without treatment, a call's chance of survival is 50%. So he has a 50% chance of dying. Now, you're probably thinking, why don't we just get some clean water and give it to a call? The thing is, when patients reach this stage of dehydration, pure clean water won't rehydrate them. Okay, so especially with the cholera toxin in there, keeping the cells continuously leaking this chlorine ion, what's going to happen is that salt concentration is always going to stay high outside the cells. And because it's staying high, what's going to happen is the water will never re-enter the cells to rehydrate them. It's actually just going to rush through your system and never re-enter, which is going to cause more watery stool and cause more dehydration because you're not rehydrating your cells. Okay, because we can't give a call clean water to reverse this condition, we need to know what we can give him. So flooding in Pakistan and the Southeast Asia, the Middle East, is actually pretty common when they have extreme rains, which means they see a lot of diarrheal diseases in these times of flooding. So a team of medical researchers from Bangladesh developed a therapy that will help rehydrate those who have these diarrheal diseases. Okay, it's called oral rehydration therapy, or ORT. So this is what a call gets when he goes to the medical center after he's been diagnosed with cholera. Okay, so ORT is an IV medication, not really medication, an IV solution that is extremely concentrated with glucose, so sugar, and salt. So we have the sodium chloride in the water, so it's going to break up the salt into the sodium plus cation and the chlorine 1 minus anion. So what's going to happen is when this is fed through an IV into the patient, it's going to directly add more sugar and salt to the patient's cells. So it's actually going to get inside the cells with that water that in the solution that's carrying them. So what's going to happen is when ORT is first administered, your salt content is extremely concentrated outside the cell and inside your intestinal tract. When you apply ORT, what's going to happen is we're going to flip the concentration gradient. So the concentration in the cells are going to have now a larger concentration of salt than the outside of the cell. And that's going to cause all the water that's there in the intestinal tract and the outside of the trail, outside of the cell, not trail, to rush back into the cell, and it's going to rehydrate it. Okay, now this is not immediately a magical cure, but it does work pretty quickly once the cells start to rehydrate. So within hours, a cow's eyes are less sunken in, his skin is rehydrating, so it's becoming firm, and within a week after receiving ORT, he makes a full recovery as if he was never sick. Okay, so that is our case study. One of the things that we are going to talk about right now is the mechanism that the cholera toxin uses to make you sick, to cause this diarrheal disease. So what does it do? Now, let me turn my little pointer on so I can follow it through. I'm going to tell you this now. This is a very important slide. This is a very important mechanism. You will be tested on this. You need to be able to explain how the cholera toxin causes diarrhea in your own words on the next exam. Okay, so we're going to walk through this. I have my little pointer here. So we're going to walk through how this happens. So now don't freak out. There's some big words here. You don't need to know all these steps. I'm going to tell you in layman's terms what's happening here. So we have our cholera toxin. This is the picture of it. It's this globby protein. It's actually 6 proteins put together. It's what we call the A subunit and 5 B subunits. Okay, so the B subunit's job is to get the toxin inside the cell. So what's going to happen is there's going to be a protein on the outside of the cell. It is called a ganglioside. I did say that right. So our toxin is going to come in and it's going to bind to this receptor. Okay, this is the B subunit's job. It's what's going to allow it to bind to it. So when the toxin binds to the receptor on the outside of the cell, it's going to signal to the cell that it needs to come in. So it's going to bring it in a process called endocytosis, if you remember that one from biology. So the cell membrane is going to form around this and make a kind of little pocket, air bubble in the cell called a vesicle, and that is going to bring our toxin inside the cell. Now, I don't remember exactly how this happens, but the toxin can be released when it's inside the cell, so it's going to be dumped into the cytoplasm. Okay, it's going to leave the vesicle, and it's going to make its way to the endoplasmic reticulum. Now, if you remember from biology, this is one of our big organelles. It's connected to the nucleus. So one of the jobs for the endoplasmic reticulum is one side makes proteins and the other side processes them and modifies them. So when the toxin enters the endoplasmic reticulum, it's going to break it up. Okay, so we're going to break the 5 subunits away, and then we're going to break this A subunit off. Now, the A subunit is what makes this toxin toxic. Okay, the B subunits aren't toxic. They can help depress your immune system, so your immune system doesn't react to the toxin as quickly, which is why individuals get so sick so quickly. But the toxic part is this little yellow guy, the A unit. Okay, so it's going to be released from the endoplasmic reticulum, and it's going to make its way back to the membrane. Now, here in the membrane, we have what we call pumps embedded in the membrane that help things travel back and forth against a concentration gradient. Okay, so the A subunit here is going to bind to one of these pumps. It's going to turn them on, so it's going to cause a shape change. Okay, and because it's a pump and it's pumping things across the membrane against a concentration gradient, it's going to require energy to do this. Okay, so the pump is going to open and it's going to start to ship out these chlorine anions. Okay, we also have a max exodus of sodium 1 plus and calcium plus ions that also travel through the channels. Okay, so we're going to pump all these chlorine ions into the outside of the cell, so into the intestinal tract, where it's going to come into contact with these 1 plus cations. So it's going to make a lot of different salts. We're going to make sodium chloride, which is the main one, and then we're going to make potassium chloride. Okay, and then because we increase the concentration of salt outside of the cell, we're also going to start to release water across the membrane. And so it's going to diffuse out via osmosis. Okay, you need to be able to explain this whole process in your own words on the exam. So you can go back and watch the slide. You can come and talk to me during flex or after class, and we can run through it if you're a little bit confused about what's going on. We also have this slide. This slide is what the book tells us is going on in here. So it has a lot of big words, but it's a good reference for you. Okay, so that's the case study. So we are going to talk about chapter 4.1 now. So chapter 4.1 is the chemistry. review for microbiology. So we're going to talk about elements, we're going to talk about different chemical bonds, we're going to talk about water and its special properties. So section objectives for 4.1 is you need to be able to describe how atoms form the molecules of life. You need to be able to describe the different types of bonds atoms can make to form a molecule. You need to be able to explain the special properties of water that allow it to distinguish between hydrophilic and hydrophobic molecules. Okay, so what are cells made of? Okay, everything is made of atoms. Molecules are made of atoms, living cells are made of molecules, which are made of atoms. Everything is made of atoms if we go small enough. Okay, so atoms are classified as the smallest unit of matter that retain the chemical properties of that element. Okay, and another property that these atoms have is that they are the smallest unit that enters a chemical combination, aka a chemical reaction. So elements are a pure substance. Okay, so they are composed of only one type of atom. Okay, elements are the type of pure substance that we cannot break down into simpler things by chemical changes or chemical reactions. So elements are the atoms, and it's the smallest form of matter that we can get. Now there are smaller things like protons, neutrons, electrons that form elements, but we can't break those down and have the properties of the elements still be retained. Okay, so all living organisms are made of some of the same elements. In fact, if we look at humans, 98% of the mass of a human is made up of six elements. So we have hydrogen, we have oxygen, nitrogen, phosphorus, sulfur, and of course carbon. We're carbon-based life forms. I would know these elements for the exam. Okay, so we're all made up of hydrogen, oxygen, nitrogen, phosphorus, sulfur, and carbon. Okay, so this is a table from our book. So we do have a few more elements besides those six that I talked about that are found in large quantities in all living things. So we talked about hydrogen, we talked about carbon, we talked about nitrogen, oxygen, sulfur, phosphorus. But we also have a large amount of sodium. We also have a large amount of iron. There's always one more and I can never remember it. But I would know the 98%, those six, for the exam. But I wouldn't worry so much about this. This is just giving you some of the properties of each of these elements. So its symbol, its atomic number, or the number of protons it has, its mass number, the number of protons and neutrons it has. We won't worry about outer shell. And it's telling us how many bonds each of these elements can form as an atom. Now, this is what is really important, and we'll talk more about this here in a second. Okay, so now that we talked about atoms, we need to talk about what happens when we start adding atoms together to make bigger things. So when we start adding atoms together, we're going to make molecules and compounds. Now, sometimes these two terms get confusing because they're very closely related to each other. So I would know these two definitions and the difference between the two. You'll be tested on this. So molecules are defined as two or more atoms connected by strong forces called chemical bonds. Okay, so molecules and their atoms can be two or more atoms of the same element. So like oxygen gas. So oxygen is found in nature as O2. So we have two oxygen atoms that are bound together with a chemical bond. Okay, so that's a molecule, but molecules can also be made up of two or more different types of atoms, so two or more elements. Okay, so we can put carbon and oxygen together, like in CO2. This is a molecule. Okay, compounds, on the other hand, are a little bit more specific. So a compound are substances that are composed of two or more different elements. So in chemistry, we have a saying that we use to kind of remember this. So all compounds are molecules, but not all molecules are compounds. So if we have a compound, it's going to be made of two or more different elements. So all compounds are going to be molecules because a molecule is just something that's two or more atoms. But say we have a molecule that's made of two or more of the same atom. So it's still going to be a molecule, but it's not going to be a compound. Okay, so in fact, when we have molecules that are made up of all of the same type of element, we actually classify them as an element still, even though they're more than one atom together. Okay, so molecules and compounds are both examples of pure substances as well, are just like elements, but with compounds and molecules, we can break them down into something simpler. We can break them down back into their individual atoms. Now, we're going to talk about some terms that we use a lot in biology and microbiology. So we're going to build upon these two definitions. And the next one we're going to talk about are organic compounds. So these are special compounds that are going to contain at least two carbon atoms and a hydrogen. Okay, and these are going to be connected with a carbon-carbon bond. So this is what we call an organic compound. So anything that's made of just carbon and hydrogen is organic. Things that are mostly carbon and hydrogen, this is organic. Okay, biomolecules are a type of organic compound that are found in cells and organisms and basically living things. So biomolecules can get really big. We'll talk more in the next few sections about the different biomolecules that make up cells. But one of the important things we need to remember when we're talking about compounds and molecules is not just what atoms make up the compound, it's also their arrangement. Because the arrangement of the atoms in a compound or molecule are going to give it its specific properties. So this is one of my favorite molecules, and it is the caffeine molecule. Okay, so caffeine is a stimulant, and its properties are all because of this ring structure and what's attached to the ring structure. So caffeine has a biomolecule that it's related to. Caffeine is an organic compound, but it's not a biomolecule, but it's related to some of the nucleic acid bases that are biomolecules, which is why it's so effective is the word I'm looking for. So effective as a stimulant, so it can wake you up when you drink it. Okay, so now that we've talked about atoms and we've talked about molecules versus compounds, we talked about organic compounds and biomolecules, we need to talk about that strong force that holds these things together. Okay, so a chemical bond is exactly that. It's the force that holds atoms together in a compound or molecule. Okay, we form chemical bonds in one of two ways. We can either transfer electrons from one atom to another in order to make sure that both atoms are going to have a full outer shell. We call this the valence shell. And once a atom gets a full valence shell, it's stable. So it's not going to want to react anymore. So we can transfer electrons. And the second way is we can have two or more elements share electrons to become stable. Okay, so depending on which way the bond is formed, we are going to classify compounds as either ionic or covalent. This is all based on the type of bonds that are inside these compounds. Okay, so we need to talk about the chemical bond that is formed when we transfer electrons. So one thing that we need to talk about is the fact that all atoms have the same areas in them. We have the nucleus where the protons and neutrons are. We have the electron cloud where the electrons are. So in ordinary chemical reactions, the nucleus of the atom, so where those protons and neutrons are, remains unchanged. This is why we call the nucleus the identity of the element, because the number of protons found in the nucleus are going to tell us what element it is, because every element has its own unique number of protons. We call this the atomic number. Okay, so the nucleus is going to remain unchanged because that's not what's participating in chemical reactions and making chemical bonds. It's those electrons in the electron cloud that are participating. Okay, so we can have these electrons participate in chemical reactions by either having the atom gain electrons the atom lose electrons or share electrons with another atom. Okay, when we gain or lose electrons, so all atoms are neutral. So when we start to gain and lose electrons, we're no longer going to have a charge of 0. We're either going to form a positive charge or a negative charge. So the formation of these charges are going to make the atoms into ions, meaning they're not neutral anymore. Now, when an atom has a negative charge, we call this an anion. So in order to make an anion, the atom has to gain one or more electrons. So if you remember from physical science, electrons have a negative charge. So when we're adding more electrons to an atom, of course its charge is going to be negative to make the anion. So conversely, if we have a neutral element and we start to lose electrons, we've lost a negative charge, which means that our atom is going to become more positive. And atoms with a positive charge are called cations. I like to remember this as cations are positive because the T looks like a positive sign if you write it out. Okay, so we care about ions quite a bit in biology and microbiology. In fact, ions had a huge role to play in the case study that we talked about earlier in this video. Okay, a lot of ions are electrolytes. Okay, they allow a charge to be present in the body. Others use energy or provide energy to move things across the cell membrane. Sodium plus sodium plus cation is really very important. We like to see sodium in the bloodstream. We need to eat a good amount of sodium because sodium's job in our bloodstream is to regulate our blood pressure and to keep it down. Other things that happen with ions is they can come together and form a ionic substance, or so a ionic compound, and these ionic compounds form crystals. When these crystals start to form in the body, they can cause some problems. If you've heard of gallstones and kidney stones, these are ionic substances that have formed crystals. Kidney stones are extremely painful because they don't break up very easily. So we need to have a good knowledge of ions to understand some of the chemistry that's going to happen in our bodies, especially in microbiology. Okay, so when ions come together, so positive charges are always attracted to negative charges. This is going to form an ionic bond. Makes sense, right? Ion is right there in the name, ionic bond. Okay, so any compound that contains an ionic bond is going to be called an ionic compound. Okay, ionic compounds are formed from a metal and a nonmetal. Okay, so we define an ionic bond in our microbiology book as the force of the attraction between the opposite charges of the ions in an ionic compound. So it's that charge difference that's holding it together. The thing is, most of our compounds that we're going to look at are going to be neutral compounds. So that means that the number of positive charges has to be equal to the number of negative charges in our compounds. So we get that overall charge of 0. Okay, so that's ionic compounds. We usually talk more about ions than ionic compounds in microbiology because our bodies are mostly water. Ionic compounds easily dissolve in water and become each of their constituent ions. So sodium salt is not usually found as sodium salt in the body. It's found as sodium plus and chlorine minus. There are extenuating circumstances, like in the case study at the beginning of the video, where there was so much of it in the intestinal tract, it started to make salt again. Okay, so that's ionic bonds. Okay, so now that we've talked about ions and ionic bonds, we need to talk about the next type of chemical bond. This is the one that's formed by sharing electrons. This is what we call a covalent bond. Sometimes it's referred to as a molecular bond, especially if you're from across the pond in, say, Europe and in the UK. So in order for an atom to transfer electrons to make an ion, energy has to be used. So it takes energy to remove an electron from an atom, and it takes energy for an atom to accept an electron. And the energy is needed to remove, or especially to remove an electron from a atom, needs to be large enough to overcome the attractive force between the positively charged nucleus and that negatively charged electrons. So a lot of times we don't have enough energy to remove the electrons, and so we're not going to form ions. Nonmetals are like this, especially the ones in groups 13 and 14 and 15. Groups 16 and 17 are a little bit more reactive, for reasons we'll talk about in a couple, I think maybe in the next lecture. Okay, so nonmetals, instead of transferring their electrons, are going to share their electrons to become more stable. So a covalent bond is defined as the attraction that forms between atoms when they share electrons, and a covalent compound is formed from two nonmetals that are joined together by a covalent bond. Okay, so a lot of our what we call the diatomic gases. Okay, so fluorine likes to combine with itself to make fluorine gas, which is F2. So what happens is when we share the electrons, we're going to get closer to having a full outer shell. So that full valence shell, we call this the octet rule because we like to have 8 in our outer valence shell. Okay, so we can see here each fluorine has 7 electrons. Okay, so when they come together and when they share the electrons, this atom has now 8, so it meets its octet, so it has a full valence shell, so it's going to be stable. This one has a full octet, okay, once it shares, so which means it's going to be stable. So we can have compounds like water. So hydrogen has 1 valence electron, oxygen has 6. So anytime you see a single electron that's not in a pair, like these two, okay, these single electrons are what are going to form the bond. So this is what is going to be shared. So we're going to bring this hydrogen over here, and it's going to be shared with oxygen's single electron here. Well, but oxygen has 6, so we have another single electron. So we need another hydrogen atom to come and share its electrons in order for oxygen to become stable. And hydrogen's a little bit weird because it can only have 2 electrons in its outer shell, in its valence shell. So once it gets 2, it's going to be full. Okay, so covalent compounds are mostly organic compounds. Organic compounds are covalent compounds. So they are going to be sharing electrons. So the simplest form of a organic molecule that's just carbon and hydrogen is going to be methane, CH4. Now carbon is a group 14 element, so it's a group 14 atom. Carbon has in its valence shell 4 valence electrons. Okay, and they're all going to be single. So each one of these 4 electrons can create a bond by sharing electrons with another atom. So we see here, we are sharing that 1 electron from hydrogen with each of our single carbon. So this is what's going to form our methane atom. We find the number of electrons in the shell. So the reason we know oxygen has 6 in its valence shell is based on where it's at in the periodic table. So it's in group 16, meaning it has 6 valence electrons. Anything in group 1 is going to have 1 valence electron. Things in group 2 are going to have 2 valence electrons. Group 13, you're going to have 3 valence electrons. Group 14, like carbon, is going to be 4 valence electrons. The highest number of bonds you can make is 4. Okay, we can't make more than we don't usually make more than 4. It can be done, but most of the time carbon is going to be the element that can make the most bonds. Okay, group 15 can form 5 bonds. Group 15 has 5 valence electrons. Group 16 has 6. Group 17 has 7. Group 18 has 8. It's the noble gases. They're non-reactive. They're already stable. And that's because their outer shell is already filled. Okay, so because we're sharing electrons and not transferring them, covalent bonds come in a few different types. Okay, so when we have a single covalent bond, so we're only sharing 2 electrons, every bond in a covalent bond is 2 electrons. Okay, so when we're only sharing 2 electrons, we call this a single bond. Okay, so that's what we see here with the hydrogen molecule right here. Okay, so we can actually share more than one pair of electrons. So when we share 4 electrons, this is 2 pairs. This is going to make a double bond. Okay, so when we share 3 pairs of electrons, so 6 electrons like we see here in the nitrogen molecule, this is going to be a triple bond. We do not have any bonds that are bigger than a triple bond. Okay, that's the biggest there is. In fact, triple bonds aren't easily made. We don't easily share 6 electrons. We mostly see double bonds and single bonds like here. Okay, when we are drawing a bond, we are going to usually use this dash notation in between the atoms that are forming the molecular compound. Okay, so what we're going to talk about mostly in this class is covalent compounds. So we have to talk about how covalent compounds share electrons. Either they share electrons equally or they share electrons unequally. Okay, so depending on how a covalent compound is going to share electrons, this is going to give certain natures to those compounds. So give them certain properties. So when we have electrons that share when we have atoms that share electrons equally in a covalent bond, we are going to call these bonds non-polar bonds. Okay, that means that each atom has the same amount of pull, so it's like tug of war, but your teams are equally matched. So the center rope of the rope stays in the center and doesn't move back and forth. Okay, it's going to be equal. Okay, different atoms have different levels of attraction to other atoms, and this level of different attractions is going to cause the differences in how electrons are shared in a covalent bond. So the stronger the attraction of one atom is going to pull the electrons closer to that atom than the other atom in the bond. Okay, the attraction of atoms, so the strength of the attraction, is dependent on the size of the atom, the charge on the nucleus, so what's its positive charge, and the total number of electrons the atom contains. Okay, so this is going to affect that strength. So in a covalent bond, a atom with a stronger attraction is going to pull the electrons in that covalent bond closer to them. When this happens, we are no longer equally sharing the electrons. Okay, so when there's an unequal sharing of electrons, this is called a polar bond. Okay, and when this happens because the electrons are found closer to one atom than the other, we're not going to make full positive and negative charges as if we would transfer them like in ions, but we're going to make a end of the molecule that's slightly positive, and the other molecule is going to be slightly negative. Okay, the one that is slightly negative is going to be the one with the stronger attraction. Okay, so it's going to be the one that's pulling the electrons closer to it. So it makes sense that it has the negative charge because the negatively charged electrons are closer to it. So these slightly positive and slightly negative ends, we call these partial charges. Okay, as I said, you can view a covalent bond like a game of tug of war. If you have two equal teams, the rope's not going to move too much, but the stronger team will, if we have a difference in strength between the two teams, the stronger team is going to pull the rope closer to their side. This is the same thing that happens with electrons. Okay, so we can then take our knowledge of polar and nonpolar bonds and we can apply it to an overall molecule. So molecules always are going to consist of at least one or more bonds. So when we get two molecules that are more than one bond, so say two or three, the overall molecule is going to be affected by what type of bonds are making it up, whether they're polar or nonpolar or a combination of the two. So a polar molecule is going to be one where there is at least one polar bond. So there's an unequal sharing of electrons somewhere in the molecule. Okay, so it's going to have a slightly positive part and a slightly negative part. Okay, but the overall molecule itself is going to be neutral. Okay, we can have nonpolar molecules. Okay, these are going to be made up of nonpolar bonds, or it's going to be made up of a molecule that has polar bonds, but the polarity of the bonds cancel each other out, making the overall molecule nonpolar. So it's not going to have any of these partial charges like a polar molecule here. A part of this is because polarity is dependent on the molecule shape, so it's what we call its geometry, as well as the type of bonds that make up that molecule. Okay, you just need to know that a polar molecule has at least one polar bond and it's not equally sharing the electrons. Oh, excuse me, a nonpolar molecule is a molecule that's going to have no oppositely charged end. There's no partial charges. It's going to be neutral overall and all around. Okay, so this brings us into water and talking about some of water's important properties. So we had to talk about polarity of a covalent compound, so whether it's polar or nonpolar, because water is the solvent of life. Water can dissolve more things than any other solvent or so any other liquid in the world that we know of. Okay, and this is because water is a polar molecule. It has polar covalent bonds in it. Okay, so here, oxygen has a stronger attraction for electrons than hydrogen. So the oxygen is going to pull the electrons to it, which is going to give the oxygen and water a slightly positive or a partial, or sorry, slightly negative, not positive, slightly negative or partially negative charge on the oxygen. And because the electrons are found closer to the oxygen, that's going to mean that the hydrogen is going to have a partial positive charge. So this difference in charges is going to be what makes this bond polar. And we have two polar bonds in water. They don't cancel each other out because of geometry. So we're going to have an overall polar molecule. And the fact that water is polar is going to be one of the reasons why it's a good solvent, so a way to dissolve things. Okay, but also one of the things that makes water cool is it can hydrogen bond, and the whole way it can hydrogen bond is because it's polar. So it's these partial positives on the hydrogen and the partial negatives that allow for hydrogen bonding. Now, hydrogen bonding happens outside of water. There's hydrogen bonds in DNA. There's hydrogen bonds in a lot of things. What you need to have a hydrogen bond is a slightly negative atom, so something like oxygen or nitrogen or sulfur. These all create hydrogen bonds, and it has to have hydrogen in the atom where the hydrogen has a partial negative. Okay, so what's going to happen is we're going to start to get intramolecular forces, so forces or bonds between molecules. So what happens in a hydrogen bond, we're going to have this slightly negative oxygen on this molecule and this slightly positive hydrogen in this molecule, and they're going to kind of form like an ionic bond. So they're going to have this force that attracts them to each other, and this is what we call the hydrogen bond. The hydrogen bonding in water is what creates the property of surface tension in water. So things like water skippers can walk across the top. If you remember from biology, we did the surface tension on a penny lab where you tried to put as many drops of water on the penny until the bubble broke. So you broke its surface tension, and we could put quite a few drops on those pennies because of these hydrogen bonds pulling the water molecules together and holding them tightly together. So hydrogen bonds are actually one of our most important bonds when we talk in biology, and they do a lot of biochemistry, but we'll talk about these as we start talking about the specific biomolecules in some of the next sections. Okay, so when we talk about water and we talk about hydrogen bonds, we need to talk about things that associate or don't associate with water. So compounds that are ionic compounds are just extremely polar molecules. So what happens is we have this saying in chemistry that like dissolves like. Okay, so polar compounds really like other polar compounds. So ionic compounds are going to associate with water, which is also polar. Okay, so when water dissolves an ionic compound like table salt, sodium chloride, we give the ionic compound the term hydrophilic or water loving because it's associating with water. We have polar um, organic compounds that aren't ionic, so polar covalent molecules. These can also associate with water because of that sameness in polarity. Okay, so hydrophilic means water loving. It's going to associate with it. Okay, so we have our sodium chloride molecule here. Water is going to come in. It's going to start to hydrogen bond. So what it's going to do is it's going to start to pull the individual ions apart in this sodium, like this cube of sodium. Okay, so we're going to have the negative parts of the oxygen that are going to surround our positive ions, and then the negative ions are going to be surrounded by the partially positive charge in this polar molecule. This is why ionic compounds dissolve so easily, is because water can tear them apart and keep them separated from each other so that they don't combine to remake the compound. Okay, if there are compounds that don't like to associate with water, they don't dissolve in water, we call this hydrophobic. Okay, so think the vegetable oil and water, you combine them together, they don't interact with each other, they quickly separate. And that's because of oil's hydrophobic or nonpolar properties. So it's nonpolar compounds that don't like to associate with water because of that difference in polarity. Remember, like dissolves like. Okay, so hydrophilic literally means water loving, hydrophobic means water fearing. So what we see here in this this example down here is we have our hydrocarbon or our organic molecule. So this is all carbon and hydrogen. It's nonpolar because both carbon and hydrogen have similar attractions for electrons, so they're going to share them pretty equally. Okay, so what we see is that it doesn't like to interact with the water molecules. So what it's going to do is it's going to clump together with other hydrophobic molecules, so other hydrocarbons, and it's not going to associate with these water molecules that are hydrogen bonding here. Okay, in fact, we can have things that use hydrophobic forces. So it uses the fact that there are molecules that don't like to attract water or associate with water and give things special properties. So an example of this would be command strips. So the command strips use hydrophobic forces, but also the toes on geckos, the hands, they have little hairs on them that use hydrophobic forces to allow the gecko to stick to surfaces and climb things like walls, which is kind of cool. Okay, so this is section 4.1. That is a long section. It's one of our longer ones. So things you need to remember, what are the elements of life? So remember those 6 from the first slide. Know the different types of bonds. Know about water is the solvent of life. Be able to explain hydrogen bonds. And know what it means if molecules are polar or nonpolar. You need to know hydrophilic versus hydrophobic. Hydrophilic is polar molecules interacting with polar molecules, whereas hydrophobic is nonpolar molecules trying to not interact with polar water.